The effect of the bacterial microbiome on the effectiveness of cancer treatments. Next slide, please. Bacterial microbiome has significant influence over the body's homeostasis and immune development/response. The diversity of the human gut microbiome far outweighs that of the human genome. We share about 99.9% of our genes, but up to 80% of our microbial genomes are different, which may serve to explain differing reactions to drugs. Next slide, please. The gut microbiome modulates immune responses and homeostasis by the production of metabolites and direct interactions with the vagus nerve. Immunotherapy is a new, promising field of cancer treatment, as cancer only survives by evading the immune system. However, it is ineffective in some individuals without much explanation as to why. Cancer drugs may also have unwanted side effects that may be prevented through a more targeted range of bioavailability, which is how much of a drug the body can absorb, and thus drug absorption. The ratio of different species and genera significantly affects treatment outcomes and can be used as potential biomarkers for the existence of cancer and trajectory of specific treatments. Interactions between these species and overall composition can be synthesized into an algorithm that can accurately determine outcomes across treatment types and thus used to inform interventions and possible treatments, such as fecal transplant therapy, probiotics, and prebiotics. The microbial difference in genera frequency between non-responsive and healthy individuals suggests there may be carcinogenic genera or dysbiosis itself is conductive of cancer. This article, in direct opposition to the next article, believes the bacteria themselves do not directly affect the drugs, but instead negatively affect the viability of the affected cancer, potentially by alerting the immune system to its existence. A high ratio of firmicutes to bacteriorhides was held in common between non-responsive cancer treatments and a multitude of other chronic disorders, including irritable bowel syndrome, treatment inherently affects not only the composition of the microbiome, but it is also inherently affected by said composition, by biotransformation. So not only do the drugs themselves affect the body, but they also affect the microbiome, and the microbiome affects the drugs themselves. So it is a loop of different effects based upon treatment. Interactions between drugs and microbes can greatly increase the bioavailability of active ingredients or reduce it to almost nothing. A specific species/genre, B. fragilis, is required for a monoclonal immunotherapy drug to actually function, providing potential use of targeted probiotics and AI in mapping out drug microbiome interactions for predicting treatment outcomes and ensuring positive results, especially when specific species are required for treatments to function. The microbiome in specific bodily systems affect the microbiomes in other bodily systems by the regulation of immune responses, inflammatory levels, and production of metabolites such as short-chain fatty acids, which can increase or decrease susceptibility to developing chronic diseases and infections. Said microbes additionally influence the expression of genes, which alters the immune system's response to pathogens beyond signaling and immune conditioning, which happens early on in life. Increased rates of certain species, such as Streptococcus, are associated with lung cancer, while greater abundances of bacterioids are associated with lessened rates of cancer reoccurrence, which means that they might be essential to have, not only to prevent cancer, but also to prevent it from happening again. Like in the previous study, a keystone species restored immune response to immunotherapy. The existence of such keystone species, ratios between species, and reactions to against drugs warrants further investigation, including the use of AI to sort data, public records, or that acquired by hospitals from patients to discern correlations. It is important to note that the fungal microbiome, which was not previously discussed in the study, in particular may have important effects on the immune system and warrants more research as it is not well understood. It is the same with how parasites affect the immune system, because they decrease the rate of allergies, but they also come at the cost of weakening immune responses to cancer and pathogens. Treatments themselves impact the composition of the microbiome and thus how the treatments themselves are absorbed. We should be taken into account when using specific treatments, including chemotherapy in particular. Narrow spectrum antibiotics may be used to remove undesired species, while broad spectrum antibiotics should be avoided due to the possibility of antibiotic resistance and negative effects on the microbiome. Bacterial microbiome is a complex superorganism which has wildly ranging effects in the body based upon diversity, species, production of metabolites, and ratio of species. The interaction between the microbiome and bodily homeostasis can be isolated into its component pieces through the aggregation of data and use of artificial intelligence, and on a wider scale too, because emergence is a thing that must be taken into account. Said information may be used to inform therapeutic interventions, such as targeted antibiotics, scans for biomarkers of cancer, as in species, just like H. pylori, or fecal transplant therapy to increase the likelihood of favorable outcomes. I am very happy to be here today. It was great working with my mentor. I learned a lot about how college works, which I've been nervous about as a rising high school senior, and especially as one who's probably going to go to a larger state school and didn't know what to think of that, to be possibly just a number, but she has taught me that can sometimes be, in a way, a blessing. And it reminds me that as a woman in STEM, I have a lot to look forward to and to look up to.